The last figure of this paper looks at a particular catalog of genes that are associated with aging, either related to sort of pro-longevity or anti-longevity, which was gathered and compiled by Fernandez et al. In Human Molecular Genetics in 2016. So in panel A and B, what this group did was basically take this catalog of genes, about 80 in humans, and I think around 63 or so that had orthologs in mice. And they basically identified the sort of lowest 5 percentile and the upper 5th percentile of these genes by median transcript length. So they took this catalog and they identified those two quantiles. And then they basically just wanted to see sort of the distribution of pro-longevity and anti-longevity genes in each of those quantiles, which is what we're seeing in these bar plots. So in the human case, this is panel A, and in the mouse case in panel B, but in the human case, we see that the shortest quantile has a basically depletion of these pro-longevity genes and an enrichment of anti-longevity genes. And the opposite is true for these longer transcripts, where we have more pro-longevity genes in humans and fewer anti-longevity genes in the longer quantile. And the same is true for these mouse genes. So this basically shows that the longer transcripts tend to be pro-longevity and shorter transcripts tend to be anti-longevity, which to me was a really interesting and an interesting finding. Next, they took these two extremes and they generated they did a gene ontology analysis looking for pathways that are enriched in one particular length extreme and depleted in the other. So here we have the networks that were generated by the gene ontology analysis, where the red nodes represent certain biological pathways that are enriched in the short extreme and depleted in the long extreme. And in blue, we have processes that are enriched in the long extreme and depleted in the short extreme. So the first thing we can really see from this panel is that there is like a very clear separation between the two pathways, with the exception of these two processes that are associated with anti-longevity, or sort of the short extreme. So that is really interesting. And to visualize this, they basically took each of these processes and they connected them based on how many genes are shared in each of those pathways. And so what we see is that in both extremes, there are very distinct processes that are systematically implicated in these, which can be systematically influenced by this transcriptome imbalance as we age, which can have really big implications for sort of health targets we can take, or targets we can look at for medicine and interventions for aging, which I thought was a very, very cool and exciting analysis.